you see my screen? Okay. So when we talk about the respiratory system, basically what are we going to be talking about? Let's see here. You want to look at if there is a system, all of the systems we have a bunch of organs that work together to perform a particular function. So the question that you would always ask are, what are the organs that are involved in this system? And what is this system's job? What is it to do? What is the purpose of this organ system? And then we'll then normally talk about some of the things that happen when the system is not working well, like the disease conditions, some of the prevalent ones, and then measures that are usually there to deal with them. So if you look at, in terms of, again, we're going to look at how we breathe, because the respiratory system is how we take in air and let out air that we don't want. So the gaseous exchanges, and then we'll look at some of the things that kind of mess up this system if it doesn't work well. We said, if you look in the pathway through which air comes, we breathe in air, then you would see that it goes from the outside. We have nostrils. Our nose has nostrils through which the air passes through. And somewhere it goes through from our nostrils, it would go through our pharynx area, and then inside there is a trachea, this cartilaginous pipe that goes down and then branches into our lungs. And when we talked about digestion the last time, we said when we needed food to be able to eat, to give us energy, and for us to be able to deal with the food after we've absorbed the food that has been digested, for us to be able to break it down to give us energy, we have cellular respiration that happens in our cells that requires oxygen to be able to break down this food through aerobic cell respiration. And so we breathe in air, which contains oxygen, so that our lungs could remove the oxygen from it and then carry it through our blood to the cells, where the cells can use it to do the cellular respiration to generate energy. And we also know that as a byproduct of that cellular respiration is that carbon dioxide is produced. So in turn, the carbon dioxide that is released from cellular respiration needs to be disposed of. So the blood carries the carbon dioxide again, brings it to the lungs in the blood. The blood brings it to the lungs, and then that exchange happens at the position of the lungs. So we have again the trachea that branches into these lungs. And then the lungs would have, if you look in terms of the trachea, the pipe itself branches into smaller pipes called this one branch here is called the bronchus. This one is the bronchus. And then the two are called bronchi. The smaller pipes are called bronchioles. And then they are going to end up in sac-like structures called alveoli, which is where the exchange really happens. And for purposes of just being able to put things in phases, we can separate all of the respiratory system into like what happens above our neck area. So call it like upper respiratory tract, and then the lower respiratory tract, which deals with the lungs. So again, I put the notes are here that if you look in our nostrils, we have hairs in our nostrils, and then within the walls in our nostrils, we have blood vessels that are very superficial in the background, which sometimes if your nose is dry and you pick it a little bit, you can even have a nosebleed because of those blood vessels that are closer to the surface. Why are they there for? The filters, the hairs are there to filter the air we are breathing so that any particulate matter is trapped in there. Some of the mucus that sometimes exists there helps also trap the particulate matter. The blood vessels closer to the surface warms the air so that because this opening goes right into our lungs, right inside our chest area. They always want that if you are like in winter when it is so, say you are in a freezing area, you don't want to just suck up cold air right into your inside your chest area. So you can't the blood vessels warm it up a little bit, and then so it passes here. Here we have this area here called the pharynx. The pharynx gas is a shared area which will come from the our nostrils to also our digestive tract. It also is behind here. So we have this thing called the epiglottis. Because this opening opens to our mouth, you can breathe through your mouth, as you know, and then, but food also comes through your mouth. So if you can breathe, close your nostrils, and still able to breathe, it means that air is coming through your mouth and some way goes through to the trachea. So this area here that is shared by both the trachea and your in the digestive tract, to make sure that when you're just breathing, you don't want food particles to go in the wrong direction. You have this epiglottis here that normally would close this trachea to make sure that food doesn't come in this direction, and then the food just goes behind here through the GI tract. Then if you come further down again, we said we have the trachea that branches into the bronchioles, the bronchi, and then the bronchioles that end up in the alveoli. One thing that you will notice in our lungs, the right lung, which is, if the way you look at it, this is the right side, this is the left side. The right lung has 3 lobes. Why would they have 3 lobes? We don't know. And then the left one has just 2. Then underneath the lungs, you have this bell-shaped, because that's when it is at rest, it looks like it's curved, like a bell-shaped a sheet of muscle called the diaphragm. The space in which the lungs are located in is a space called the pleural cavity, and the walls are kind of smooth, so that they're called the pleural membrane that lines them, so that when you breathe in and out, the lungs will expand and rub against those walls and not causing any too much friction. So we're going to look at what happens to these muscles and to the lungs when we are breathing in and when we are breathing out. Then if you have a bigger picture here that looks at the upper respiratory tract again, just describing all of the parts that I told you here. And then if you go here, sometimes we have situations where when you cry, your nose runs, because we have, our tear glands sometimes would make liquid to come through here to get to our nose sometimes, and then you have, out of a sudden, you are crying, then your nose is running, and then all of that. But you have there's an opening in the back here, throat that has some of these connections. But the point here is, through our nostrils, we breathe in air. Our nostrils, we know we can also smell things because we have nerve cells that are able to detect smell. And then we said we have the blood vessels that are superficial there that would allow for warming of the air as we breathe it in. Then I think I put some small extra notes here to talk about all of that. Then the opening again is much more revealed here. And then the other thing that I will also mention here, when we're talking about our immune system, we talked about, because these openings from the mouth, from our nose comes here, we have our tonsils here, which are locations where we have white blood cells, being that, within this proximity, should something that you are eating or something that you are breathing in tends to cause a microbe try to create an infection in this area, because of the proximity of these tonsils here that have white blood cells, they're able to start intervening easily and quickly. The larynx is our voice box, say this triangular structure here, that when air passes through, it makes noise, and that is how we are able to speak. Then you talk about the tongue here, but this is anatomy of it. But in terms of the respiratory system, let's focus on this area. The upper respiratory tract comprises of the nose, your pharynx, and your larynx. Meanwhile, the lower respiratory tract is made of the trachea, the bronchial tree, meaning the bronchi and the bronchioles, and then the lungs. I mentioned, if you look in the trachea here, if you look at how the walls are under the microscope, you will notice that they have this hair-like structures called cilia. It's almost like those carpets that used to have like threads sticking up. It's called cilia. And then embedded in them, you have these goblet cells that secrete mucus. So the trachea is lined by cilia, within them goblet cells that secrete mucus. Together, if you are in a dusty area, for example, and then you are breathing, if particulate matter comes here, this cilia and the mucus traps it, because these cilia are capable of moving. When they trap it, then you have this sensation that happens, and then sometimes you will cough. If you have been in an area that is very dusty, sometimes when you cough, the sputum that you come out is brown. It's thanks to the mucus that has trapped some of the dust, and then when you cough it up, it comes out by like a reflex gagging. You come out, and then when it gets into your throat area here, you have a decision to make, whether if there are people around, you just kind of swallow it, and then it goes through your esophagus, and then you goes to your stomach. Or if there is nobody there, then it's okay, and then you can cough it out and actually spit it out. Again, so if you notice now, so you imagine that there are these pipes that comes through our nose connected through this opening that comes from the outside right to the inside. So if you look at how this structure is, it's not just a naked pipe, but the walls of it are designed in such a way that to make it function properly, without minimizing the chance of particulate matter coming in, you have cilia, you have mucus that is secreted by these goblet cells. I said the bronchi continue until there are small bronchioles that are about 1 millimeter in diameter. So these ones here are like 1 millimeter in diameter. They end up in sac-like structures which we will see in the next diagram, that are called alveoli, which is the site for gaseous exchange. I already mentioned that the right lung has 3 lobes, and the cavity in which they exist in is called the pleural cavity, surrounded by the pleural membrane. Let me see whether this place here is an animation to show you how our lungs work. Let's see. Let's give this a try. Let me know if you can hear. Just give me a thumbs up if you can hear the sound. Okay. I'm going to kind of, all right, let's start to play this here.
External and internal respiration. When we talk about respiration, we are referring to the processes involved in the exchange of gases. In our bodies, gas exchange occurs between our lungs and the atmosphere, and between the blood and tissues of the body. The exchange of gases between lungs and the atmosphere is referred to as external respiration because gases enter and leave from the environment outside the body. The exchange of gases between the blood and tissues is called internal respiration because gas exchange happens between the cells internally. To better understand these processes, we're going to follow the flow of gases in a normal breathing cycle and examine what happens to both carbon dioxide and oxygen as you breathe in and breathe out. We will start with oxygen. As you breathe in, oxygen-rich air enters the lungs. Once inside the lungs, this oxygen-rich air makes its way to tiny air sacs called alveoli. Each of these alveoli is surrounded by a network of tiny capillaries. In the capillaries, specialized blood cells called red blood cells, or RBCs, are returning from the tissues of the body, where they have dropped off their previous supply of oxygen. Because the red blood cells aren't carrying oxygen, the partial pressure of oxygen inside these cells is lower than the partial pressure of oxygen in the alveoli. This difference in partial pressure causes the oxygen to diffuse from the alveoli into the plasma of the capillaries and then into the red blood cells. Once in the red blood cells, the oxygen binds to hemoglobin, forming oxyhemoglobin. A note here about partial pressure. Gases exert pressure, and the amount of pressure each gas exerts is called its partial pressure. So when considering diffusion of gases, you can treat partial pressure the same way that you treat any other concentration difference. A gas will diffuse down its concentration gradient from a higher partial pressure of oxygen to lower partial pressure of oxygen. Thus, during external respiration, this means oxygen will diffuse from the alveoli into the plasma of the capillaries and into the red blood cells. The oxygen-rich RBCs then return to the heart by way of the pulmonary vein. They are then transported using the systemic circuit of the circulatory system to the tissues of the body. Once the RBCs reach the tissues, internal respiration begins. The partial pressure of oxygen in the RBCs is higher than that of the surrounding tissues, which are using oxygen for cellular respiration. The oxygen, therefore, is released from hemoglobin and diffuses out of the capillaries and into the cells of the body. Now we'll follow the transport of carbon dioxide gas, starting with the RBCs that just dropped off oxygen to the tissue cells. At about the same time as the oxygen is diffusing outward, carbon dioxide diffuses into the RBCs. However, unlike oxygen, which was largely transported by hemoglobin, only a small amount of carbon dioxide gas binds to hemoglobin, forming carbaminohemoglobin. Most of the carbon dioxide combines with water to form carbonic acid, H2CO3, the same chemical found in soda. The carbonic acid breaks down quickly into bicarbonate ions, HCO3- and hydrogen ions, H+. Bicarbonate ions diffuse out of red blood cells into the plasma and are the major transport form of carbon dioxide in the blood. The globin portion of hemoglobin combines with excess hydrogen ions produced by the overall reaction and becomes reduced hemoglobin. Once again, this is referred to as internal respiration because it's occurring between the tissues and the RBCs. The systemic circuit returns the oxygen-poor blood to the heart. From here, the blood is sent on to the lungs via the pulmonary artery. In the lungs, the gas exchange process is basically reversed. Bicarbonate ions diffuse into RBCs and combine with hydrogen ions from the reduced hemoglobin formed during internal respiration to form carbonic acid. Carbonic acid is broken down by an enzyme called carbonic anhydrase, forming carbon dioxide gas and some water. Carbon dioxide also dissociates from the carbaminohemoglobin formed during internal respiration. This carbon dioxide gas then diffuses out of the RBCs, across the capillaries, and into the alveoli, from which it is eventually exhaled into the atmosphere. Notice that both internal and external respiration largely rely on the process of diffusion. While the physical act of breathing requires energy, the process of gas exchange or respiration does not.
Cool. So you have, I put these animations here so that when we talk about them, or when you have a better way of imagining or visualizing what is happening. And if from our slides here, we said each lung, there are about more than 3 million of these alveoli located here. And this is the location where gaseous exchange happens. And when we talk about this exchange again, as you see, when we're talking about movement of material, we talked about diffusion. So this is an example of where you think about it, how does carbon dioxide get in from the air that we are breathing in into our blood vessels? And how does oxygen get from the air we are breathing in into our blood vessels? And why does carbon dioxide that is being carried in the blood that is brought to the lungs, how does it leave that blood and get out to the, through our trachea back for us to breathe it out? So this video kind of illustrates that. And the mechanism, again, is by diffusion, which involves a higher concentration of a particular kind of molecule in one section compared to the other section, such that the molecules will move from where they are in higher concentration, where they are in higher partial pressure, to where they are in lower partial pressure. So we talked about inspiration and expiration. Inspiration, the active process by which we inhale, bringing air into our lungs. Expiration is the opposite. And then in terms of what is happening to our lungs and to the diaphragm, as I was mentioning, during each of these phases, our ribs have muscles in between them called intercostal muscles. I mentioned to you about the diaphragm. So I said volume of the thoracic cavity and the lungs would increase when we are bringing in air. When we are bringing in, your chest usually rises up and expands. So when it is rising up, the intercostal muscles have contract, bringing your rib cage up, and then the diaphragm contracts, from getting it from its bell shape to a flattened state. When that happens, the thoracic cavity almost like increases. It increases. So the lungs are capable of expanding. And when they expand, the partial pressure of air outside is higher than the one inside your lungs. So air would flow into your lungs. Air flows into our lungs. We don't go around, like when we go around, you breathe normally without being conscious about it. You just kind of keep breathing as we are talking. You don't go, no, you don't do that. We just walk around and do that. So this idea of breathing in and out is almost like a passive process. And the air moving in is just because of the differential. There's more air outside than there is inside. When we have increased our thoracic cavity by our intercostal muscles contracting and our diaphragm flattening out, so there's more space. That air will rush inside. When the air rushes inside through the trachea, the bronchi, the bronchioles, and right to the alveoli, then you have that gaseous exchange happening there. Now, in the opposite directions happen, because when you like breathe in, it doesn't stay like that. After a while, you need to breathe out. Then the muscles relax. When the muscles relax, the intercostal muscles relax, the diaphragm relaxes. It's almost like it narrows the space again in our thoracic cavity, forcing the air that is inside the lungs now to be pushed out. To be able to understand the health of our lungs, how much air we are breathing. So they have these things that people who do work with about who take care of our lungs, they try to understand. They have these volumes definitions here, like the tidal volume, which is the smallest amount of air that usually moves in and out each time we breathe. Because when we breathe, there's some certain amount of air that comes in, a certain amount of air that goes out, and then there's vital capacity, the maximum volume of air that can be moved in, plus the maximum amount of air that can be moved out. So when you breathe normally, and then I said breathe in more, then out of a sudden you forcefully or intentionally breathe in more, that maximum amount of air that you can breathe in, and then when you relax, the maximum amount of air you can breathe out is called the vital capacity. Then we talk about the inspiratory and expiratory reserve volume, which is said the increased volume of air that you move in and out. So if you imagine that you're breathing in normally, then I say breathe in, and then you inhale more, and then I say breathe out, you breathe out more. That difference between the amount of air that you're breathing normally, and then the one that you breathe in extra, and then the one that you breathe out extra, is called the inspiratory and expiratory reserve volume. Our lungs always have some amount of air in there. In that way, they don't collapse. And that amount of air that stays inside our lungs to keep them open, because we don't normally, we are unable to just push everything out. That amount of air that stays there is called the residual volume of the air remains, called the residual volume. And being able to measure all of these different volumes, it can give a good sense of how well our lungs are functioning. So we have instrumentation that they use to be able to make this determination. So the spirometer, they put the close your nose, and then you suck stuff up. Then it shows up on this machine like this, and then the information is captured to be able to determine your lung health. So now when you look at, okay, what controls? When we breathe, normally we are, as I mentioned again, that we breathe going around, not saying, okay, breathe in, breathe out. It just kind of happens without us really paying attention to it. I said the respiratory control center in our brain is located in an area called the medulla oblongata. It would send impulses through nerves to these muscles that we mentioned, the intercostal muscles and the diaphragm, making them to contract. And we do that, it does it without us being aware of it. If everything is working well, we breathe without knowing. This happens in very young, even in babies. Sometimes we have this situation called a sudden infant death syndrome, where babies, they lay the baby to go to sleep, and then out of a sudden, the baby doesn't wake up, just kind of sleeps and dies in their sleep. One of the hypotheses that they are believing is responsible for this condition is that this medulla oblongata that normally communicates to our muscles, and in the baby, sometimes it might not develop very well, and the communication might not be working very well, and then sometimes the signal ceases, and then the baby dies in their sleep. So what would control when we breathe, our breathing goes faster or slower, or is just maintained? From the video that you saw, when the levels of carbon dioxide are high in our blood, the carbon dioxide will combine with the bicarbonate ion to form carbonic acid. And when the increase in acidity measured by the pH falls within a certain threshold, as our blood doesn't, there's usually a happy pH where our blood stays in, and the more carbon dioxide that is present, then it will make the pH to go down more, to make it more acidic in our blood. Then that pH sensor would then be detected by our brain, and then the message will go from the brain to these muscles and say, please do something faster, breathe more, to be able to make sure that the carbon dioxide is let go in the lungs, and then more oxygen is brought in. And what they said, what senses the pH conditions again are receptors that we find like in the carotid and the aortic in our blood vessels that are able to sense this. I said both two sets of chemoreceptors sense the drop in pH. One set is in the brain and the other is in a circulatory system around the carotid, our carotid and the aortic bodies that are found in these blood vessels. So we mentioned the idea of what is happening at the level of the alveoli, talking about partial pressures. Partial pressure, again, if you want to look at it, forgetting the word pressure, pressure is usually defined as the force that an air exerts on per square inch was in an area. But in this case, they are saying, what is the relative concentration of carbon dioxide or oxygen, either in the lungs or in the blood? And wherever the concentration is higher, the molecules of carbon dioxide would move to the other direction. So, for example, if the concentration of carbon dioxide in the air is less than the one that we have in our lungs, meaning that in our blood vessels, meaning that if the blood has more carbon dioxide in it, it would flow from the blood to the lungs. And same thing for oxygen. We said, okay, the way in which oxygen is transported in the blood, simple, has a hemoglobin, which we see in our red blood cells, would bind oxygen to form oxyhemoglobin, and then it will carry it. The red blood cells would take it to the tissues where exchange would happen. The oxyhemoglobin releasing the oxygen to now go to the tissues, to the cells where they need the oxygen. And while it is doing that, the carbon dioxide does the difference. Carbon, I said, binds with the hydrogen, binds with bicarbonate ions to form the carbonic acid. Then it would go now, when it gets to our lungs, this enzyme breaks it down to form water, and then carbon dioxide, which escapes in our lungs. And I think you saw this picture that was being illustrated in the animation that you saw. So this is a picture that I would say familiarize yourself with and be able to explain. Blue coloration usually indicates blood that is higher in carbon dioxide and lower in oxygen. So you could take the circle again from anywhere. You could start from the lung, from the heart. From the heart, the pulmonary artery would carry this blood that is rich in carbon dioxide to the lungs. Why? Because it is here where the carbon dioxide will be released. The carbon dioxide is being released from the blood to the extracellular matrix and then to the alveoli space. And then while this is going on, oxygen, if you want to consider oxygen on the other hand is moving. Since the oxygen in the air, alveoli space is higher, the oxygen will move from here into the blood and is bound by hemoglobin and is carried along and brought back now to the heart. Then it goes through systemic circulation At the tissue level, exchange happens again, and then the whole system continues. We talk about how oxygen and carbon dioxide are carried. Now, sometimes there are some conditions that would happen because we mentioned all of these things that could prevent infections can happen from happening, but sometimes they do. And then when they do, sometimes our sinuses, remember we're talking about the sinuses that we saw that had, sometimes they will get blocked. Our tonsils will get blocked, and then sinuses would also get blocked. And then sometimes we have infections of our middle ear. Sometimes we have infections or inflammation of and infections of the larynx, causing all of this. So you have here rhinitis, where our sinuses, and then pharyngitis, then our laryngitis, all of this, and you exhibit them sometimes having what you have pressure. If your sinuses are blocked, then you have some tension around your nose area, your forehead here. And then if your ears have an infection, there you have a situation, then you have pressure here, the larynx here, and then your voice is kind of messed up. Some of these things, symptoms are usually associated with you have like a cold or a virus infection or some kind of infection. And then these ones, as you can see, are the things that would affect the upper respiratory system, because we said the upper respiratory system is any part of the respiratory system that is from our neck up. But meanwhile, again, the lower respiratory system that has to do with our trachea, our lungs themselves, we have diseases like pneumonia, which has fluid buildup, tuberculosis, where it's a bacterial infection that forms capsules with pus inside, and then you have pulmonary fibrosis, where you lose where you have fibrous tissue building, connective tissue building up, making the lungs not to function properly. Emphysema, which is when your alveoli are messed up meaning they used to hang like small balls of grapes, but now out of something, the walls break down and then reduce the surface area, which makes your ability to do gas exchange much more inefficient. And then asthma, which would have situations where the walls of the bronchi gets irritated and inflamed. And when they inflame, that means they get bigger. Then this passageway gets narrower. And when it gets narrower, as you breathe in and out, because the passage is small, then you can hear like sound coming in. They call wheezing people who have asthma. And then sometimes you could have a situation where the cells of our lungs starts dividing in an uncontrollable manner, where you have lung cancer. And I put some pictures here to kind of illustrate which one is happening where. So you have the alveoli here, pneumonia. Here you have an infection that causes fluid to build up. And when this fluid is built up, it messes up the ability for gaseous exchange to happen properly. Fibrosis, you have connective tissue builds up again, reduces the ability of the lungs to be flexible, expanding and relaxing to do what they are doing properly. Tuberculosis, you have this tubercles that are created in here, and when they also do that, they mess up the elasticity of the lungs and then mess up the ability for them to do gaseous exchange properly. Emphysema, you see the walls here have collapsed. Then instead of smaller alveoli, but now you have a big hole here, which means the surface area is reduced. Because if you look at this, if this is round, this is round, this is round, the surface area is increased. But if you are just looking at this one, the inside here collapse, and then you have a big hole here, and then the surface you're talking about is only the one on the exterior here, then the surface area is reduced. Emphysema, so this in the bronchi and the bronchioles, they swell up, inflammatory response, they would swell up, maybe something would sense, and then we're talking about how histamines are released by mast cells to cause, and then it causes the tissue to swell up, and then when it swells up again, the narrower this opening is inside here, the more each time air wants to go through, you start hearing sound. Some of the things that could be implicated in or habits or things that will contribute to the health of our lungs would be either lifestyle usage of things like tobacco, or staying in area that has a lot of dust or smoke particles. Some of these things could damage our lungs. Some of them would even cause cancer in our lungs. People who have lung cancer, for example, have a survival rate that is not very good. And we know that if you smoke, for example, it is not only your lungs that might be affected, because some of the things in the smoke that you inhale can even be carried in the blood and cause issues in certain other areas. And it's worse even for people who are pregnant, because it can affect the developing fetus. And for bystanders who do not smoke, again, if you get even exposed to this a lot, you can also have an effect of partial the smoke that some other people are smoking. And even when, for example, we had fires in Los Angeles areas and all the smoke that was happening, we have oil refineries that have sometimes flare up, and then things get released. All of those things that kind of put things that are not supposed to be in our air, and then we breathe it in, they have a potential of causing us issues. And many a times, some of the habits that we have sometimes come into conflict with our freedoms. And if people say, I want to smoke, and then somebody says, you shouldn't smoke, then sometimes you go and I would tell our legislatures to pass laws to say people should not smoke. It kind of infringes on people's freedom. I think we are free to do things sometimes that are not in our best interest, but sometimes that being able to do some of those things, they interface with the interests of others. Like when we're talking about smoking, if you smoke in your basement, well, it might be good enough that you are not sharing the smoke through secondhand smoke to other people, but the impact on other people does not just end there. The impact would be because, for example, we have an insurance system where the more sick people there are in the pool that is being insured, the higher the premiums would be. So sometimes it is not just infringing on people's rights when we say that people should have habits that would keep them healthy as a society. It is because sometimes if people carry out unhealthy habits, they can get sick, and the more sick people there are again in the insurance pool, the more the premiums, which is how much we pay for insurance, for some people who are healthy, is also going to increase. So sometimes there's always this controversy whether should we ban certain of these habits. If we are banning smoking, we know that people who drink soda that has a lot of sugar is not good for us. Should we also ban sodas, or should we only ban cigarettes? So all of those is usually a talk of war in terms of our discourse in society. But as members of the society, I think it's important for us to always consider this and weigh in appropriately. But for self-care, things like smoking, we know that there is no benefit for us. There's no value, except somebody knows of any benefit of smoking cigarettes. If you know that, you can share, but there's no benefit for it. People don't only smoke cigarettes, they smoke other things, like smoke weed. We know that cannabis, for example, So some of it, we still have some of the things that are not desirable inside, but we also know that things like cannabis, they have discovered that there are molecules that are present in there that might be ameliorate some pathological or conditions that people have. But we need to know more about that. And we know that if there is actually something in cannabis, which is weed, that benefits us, and it could be delivered through inhaling, because we have aerosol things that people inhale, meaning as a drug delivery method. One of the challenges that cannabis has is that when you take like an inhaler, they tell you to take 2 puffs, and each puff contains this milligrams of something. In cannabis, it's difficult to even tell how much quantity of material you are taking into your lungs or into your system. So it is something that on a scientific level, they need to develop more to be able to understand it and know whether if it has a real therapeutic value, how they can create doses for it to be able for people to benefit without having negative repercussions from using it. I think this is all I have here. Does anyone have any question? I'm going to stop sharing for a second. So if you look logically, I said, for example, we have the systems to do things. We have the respiratory, we talked, I think we did digestive system already. We've done the circulatory system. And then now we've talked about the respiratory system, whose bigger goal is to be able to manage our need for oxygen and be able to eliminate the byproduct carbon dioxide that we produce as a result. So then now if we have talked about the digestive system, and then we've talked about the respiratory system, and we've talked about the circulatory system, then we know that the reactions, that the cellular activity that happens within our cells produce carbon dioxide. We've seen one way to eliminate it. There are certain other things that are produced as a result of the cellular processes that happen in our body, in our cells, the metabolic processes, I would say. So sometimes we have things like from the digestive process, certain things are eliminated, some things are absorbed into our blood, and then sometimes we need to, when we take medications, we have to sometimes get rid of some of them. So let's take a, before we even get to the skeletal system, let's look at the urinary system, because now we have oxygen being brought in and out. We know cellular activity happening. We know one of the byproducts again is carbon dioxide. Then we know how we've seen that how that is released. Let's look at how some of the other metabolic waste, so waste that come from the other reactions that happen within the cell, how those are eliminated. Let's talk about the urinary system and the excretory system before we talk about the skeletal system. I'm going to share my screen really quickly, and then we go through that as well. Where are we? Yeah. So they call it the urinary system and the excretory system. The excretion is when we urinate, we pass some of the waste that have been produced as a way of excreting them. So I put here, I said, when the approach here is very similar. We're going to look at the parts that make up the urinary or the excretory system. Look at the big parts and look at the microscopic parts that are involved. And we know that then we'll look at the mechanism that happens to be able to remove the waste and how the waste are normally then eliminated. We know that in the urinary or excretory system, the big players would be our kidneys. And as our kidneys go to remove some of the waste, the way of eliminating that waste is through urination. So the question is, okay, how do the kidneys, how do the kidneys actually, we see them like big structures. You saw them when you dissected the rat. So how do they really function? We'll look at the functional units, filtration units that are within the kidney that make this happen. So if you look at the organs that make up the urinary system, I put here you have we have two kidneys. We can function with one, but we have two. They are bean-shaped structures that are located on the posterior side, almost like in the middle section of our backs. Above them, we have these structures here that are called adrenal glands. You would also know that when we talk about the kidneys, we usually refer to them as renal. So these glands here, they are called adrenal, almost like adjacent to the renal kidneys. Then you have these connections here, the blue one and the red one. The blue one is the blood vessel that is carrying material, carrying stuff to the away from the kidneys, and the red one is the one that is bringing blood to the kidneys. So you have blood being brought to the kidneys and blood leaving the kidneys. Okay, so the blood is brought to the kidneys to do what? To do the filtration and then excretion. And the product that leaves in the form of urine, that is called. So there's a connection from the kidneys that does the filtration and the bladder, which is down here, where the urine is stored. And when the urine fills up in the bladder, then impulses are sent for the bladder to control, and then we pee. So the connection between the kidney and the bladder, you'll hear the word ureters. Without an H here, ureters. And then you would see that the connection from the bladder to the outside is very similar to this word ureter, but it has an H, urethra, passing from the bladder to the outside where we pee. For the women, the urethra is shorter, and for the guys, the urethra is longer. And sometimes you would notice that because of the shortness of the urethra in females tend to, meaning that their bladder is much more closer, the connection from it to the outside is shorter. Sometimes they tend to have more bladder infections than guys. Now, so in terms of general functions of the kidney, what do the kidneys do? The kidneys excrete metabolic waste. And I put the word again, metabolic waste. Actually, let me put this on a bigger slideshow scenario so that we can see better. Okay. They produce metabolic waste. So when we say metabolic waste, I say metabolism is a sum total of all the reactions that happen within our body, right? And when you say within our body, that's probably happening within our cells. So waste that are generated from those reactions would be referred to as metabolic waste. And then you now would need to fill in the blanks. It's what are the waste and how does the filtration to produce this waste, how does it happen? Then the other function of the kidney is to maintain our blood pressure. And when they do so, they maintain the water-salt balance. The amount of salt, how many ions that we have in our tissue fluid, in our blood, would inform the blood volume. And the blood volume would also affect the blood pressure. So to make sure that we have certain ions like sodium ions, like potassium ions that are present, they always keeps to make sure that we have them within a certain range to make sure that they only bring in enough liquid to have enough blood volume, not to create a spike in blood pressure. We will also see that it also secretes hormones. it will secrete hormone and erythropoietin. We'll see that renin will have to do something in controlling this water salt balance, and then erythropoietin, we mentioned before in our last lecture, is involved the production of red blood cells. So erythropoietin is secreted by the kidneys. It will go now to our red bone marrow and make them to produce red blood cells. I said it also reabsorbs filtered nutrients and converts material to vitamin D. So let's look at the excretion phase. What does it excrete? A bunch of nitrogenous waste. It means that waste that contain nitrogen. And some of these wastes would be urea, which comes as a result of the breakdown of amino acids in the liver, uric acid, which is also produced as a result of the breakdown of nucleotides, and creatinine, which is made by our muscle cells from the breakdown of creatinine phosphate. These are the nitrogenous waste that the kidneys have to selectively remove from our blood. I said they maintain water salt balance and acid balance, the pH. So I said both are homeostatic mechanisms, because why? We need our blood to be at a particular desirable pH range within have a certain amount of acidity. And we also needed to have a certain amount of salt concentration to make sure that the water, the volume of the blood is in a way that the blood pressure is within an acceptable range. So I said the water salt balance helps to maintain blood pressure. When the kidneys excrete hydrogen ions and reabsorb bicarbonate ions, they can make the pH to increase if it was going to acidic. I said this acid-base balance helps to maintain blood pH, which is desirable to be around 7.4. So I said the normal pH is again for blood within this range. If it goes higher than this range, we said it's more basic, we say alkalosis. And then when it is lower than 7.4, we said it's going to be acidosis, means the pH is low. So sometimes when they're looking at your blood, when you give blood, they want to analyze it, they will look for all of these conditions. I say the pH of fluids is maintained through several mechanisms, the acid-base buffer system, and then through the respiratory center, because remember, if we have higher carbon dioxide because we are not breathing air properly, the blood will become acidic because carbon dioxide will combine with water to form carbonic acid, and that acidity drops the pH. So the respiratory system has a way to play in terms of eliminating carbon dioxide to make sure that our blood doesn't become so acidic. Then we have buffers that help to make sure that they also manage the pH. I say buffers are solutions or substances that can take up excess hydrogen ions or excess hydroxide ions. So I said when hydrogen ions enter the bloodstream, this reaction occurs. They would bind to bicarbonate ion to form carbonic acid. When there are more of these hydroxyl ions, which is in this condition, the alkalosis condition, the hydroxyl ions will bind with the carbonic acid to form the bicarbonate and water. So we have this situation where we have this acid and this bicarbonate ion all interacting with the hydrogen ions or the hydroxyl ions to do what? To manage the levels of OH and to manage the levels of the hydrogen ion. More hydrogen ions account for acidity, more hydroxyl ions account for basicity. I mentioned secretions of hormones. I already mentioned that renin is secreted by the kidneys. What does it do? It makes these adrenal glands that I was showing you that are above each kidney to secrete aldosterone. Aldosterone will control how much sodium is released or retained, which would in turn regulate how much water comes in. Erythropoietin, I already mentioned that it goes to stimulate red blood cell production. You can look at this later on. And then I said, so if you look at the process of filtration, some material gets some desirable material gets filtered out, but they are usually reabsorbed. I said the urinary system is responsible for reabsorbing some of the filtered nutrients, one of them being vitamin D molecule. And the more vitamin D we can retain, the more we are able to absorb the calcium that we eat in our diet. So let's look at the structure of the kidney. We've seen that there are two large organs, right? Two bean-shaped, slightly on our back and our posterior. But if you're going to slice one up, you have a diagram that will have like, if you slice it up, you take beans have two cotyledons, like two sides, which you can break them open and then it becomes like two sides. So if you can imagine that a kidney is bean shaped, if you just slice it transversely, then you would have an exterior part or what is at the edges here that is called the renal cortex. Then you have a middle part here that is called the renal medulla. And then you have these structures here that kind of project here and then go to this opening here that they call the renal pelvis. And then this renal pelvis now connects this opening here, goes into a tube that goes down, that tube that we're referring to as the ureter that goes down to the bladder. Blood is brought by the renal artery. Blood leaves the kidney through the renal vein. Okay. So again, to look at it in more the same structure that was diagrammed there, this is a picture of a real kidney. When you slice it open, it will look something like this. The cortex area, the medulla area, and the pelvis area. Now, if you look now, how does the filtration happen? It's not like one big filter system, but rather a small of microfiltration units that are located in the kidney. And these filtration units are referred to as nephrons. So this is one nephron, this is the other nephron. There are many millions of nephrons. So the filtration happens here, the reabsorption happens here. The produced waste is carried in a liquid called urine that now comes through from each of these filtration units and comes here to the pelvis, which again is this part that would now, so it's like the nephrons here, the nephrons here, the nephrons here would connect here to this opening here, which is the pelvis. The urine drips out and then collects from here, goes down here through the ureter to the bladder. It's a bigger picture here of the nephron. So I said for one kidney, there are millions of nephrons. So they make them very efficient. It's not like one system that it falls out, we need to repair it, the kidney is not functioning. But if one nephron is messed up, we have a bunch of them that also keep the process working. So again, you have I said blood is brought here through the renal artery, and then the blood comes here, and it goes, the blood vessel comes and it branches now into this. It's almost like you took a spaghetti. It's a long tube the size of a spaghetti, and then you kind of bunch it up, and then from that bunched up again, a tube leaves from it. So the renal artery, they call it the afferent arteriole, comes here, it bunches up in this structure here to form what is called a glomerulus. And then now it leaves from this bunch of glomerulus through an efferent arteriole. See, afferent, 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 then leaving efferent. But in this structure right here, the glomerulus, a lot of activity happens here, a lot of filtration happens here. Certain molecules you are going to see would leave from here because the walls of this bunch of here are small enough for certain molecules to be able to come through. So when those molecules come out here, they go through a cup-like structure in this capsule here called the Bowman's capsule. And then the liquid moves through here in this tube that kind of folds here. They call it a convoluted tubule. And then it comes here, it loops down here in this U-shaped structure here called the loop of Henle. And then it starts ascending. Again, it folds up here, convolutes here, and then it connects here to this collecting duct, which you will see from this nephron, it connects here. From another nephron, it's going to connect here. From another nephron, it's going to connect here. But this is a collecting duct. It is this collecting duct that would go to the pelvis. And then from the pelvis, it joins the ureter. So I put the notes here for just what I was describing to you. I said the glomerulus is a knot of capillaries inside the glomerulus capsule, sometimes called the Bowman's capsule. It has pores to produce a filtrate, meaning that the blood that is being brought here through the afferent arterioles, because of the pores that are here, certain material can come out. Because of this folding here in this structure, this one is closer to the glomerulus. They call it proximal convoluted tubule. This other folding here is far away from the glomerulus. They call it distal convoluted tubule. Then we talked about the collecting ducts. So in the process, when the material or when the waste is filtered from the kidneys, it is carried out as urine. So the process of urine formation involves 3 steps. Glomerular filtration, the filtration that is happening at that glomerulus. Reabsorption, as we are going to see, for some material that was filtered out, but that is still desirable by the body. And then secretion, the rest of the other material that is not reabsorbed, that needs to just continue as a filtrate, and that will form urine. This is a bigger structure of it. So we have, again, the blood vessel coming here. The afferent arteriole brings blood here under pressure. Some of the material filters out. If you blow the structure here, the glomerular capsule, the things that come out through the pores that are on here would be some water would ooze out, urea will come out, some glucose will come out, amino acids will come out, uric acid will come out, some salts will come out. So if you look here, glucose, yeah, it's good, it's still needed by the body. Urea is a waste. Amino acids are still required by the body, but they come out because just in terms of pore size, they are able to move out due to the pressure. Uric acid is a waste that needs to be eliminated. Salts, yep, some salts are pushed out, but we don't want to remove all the salts. Now, depending on the amount of salt that we need back in our blood, some of it will be reabsorbed. So the first step of urine formation would be this filtration that happens at the level of the glomerulus. So imagine that all of this stuff was filtered out. Now it is now flowing through this proximal convoluted tubule. It flows here, flowing this way, flowing this way, and then ultimately will connect here to this connecting duct. But before the liquid that was moving through here gets to this connecting duct, you have this efferent arteriole here that goes and divides and kind of creates a mesh over this proximal, the looped region and the distal convoluted tubule. So the blood vessel just kind of creates a web around here. And then afterwards reconnects back when it is leaving this this area, these convoluted areas. What is happening in this? Why do these blood vessels that were leaving the afferent arteriole create this mesh? They create this mesh so that, again, if some materials can be reabsorbed back into the blood vessel, some amino acids will be reabsorbed back, glucose will be reabsorbed back, some salts will be reabsorbed back. So I said tubular reabsorption, nutrients and salt molecules are actively reabsorbed from the convoluted tubules into they call it the petubular capillary network, and water flows passively. So there is active reabsorption, this reabsorption of material back into the blood vessels here. Amino acids, glucose, because they are still needed by the body. Some salts, depending on the amount that is needed, but the rest of it will continue now for the tubular secretion. So you will see some drugs like penicillin or some other things get secreted at this level. The kidney helps in their secretion after the drugs have been metabolized. And then at the end here, what will be coming out into the pelvis area that is in the urine will be water, urea, uric acid, some salts, some ammonium salts, and creatinine. Again, the blood here is under higher pressure and the blood leaving here is under lower pressure. The higher pressure would cause these various molecules to come out, and then they will continue. They're called they're called the glomerular filtrate, right? The glomerulus filtrate. That they will continue now through the proximal convoluted tubule, right through the loop of Henle, the distal convoluted tubule to the collecting duct. Again, I put here the things that, first of all, when we say the things that come out are all of this, but the things that do not come out are like blood cells and platelets, and plasma proteins. So when they take your if they take your pee, when you do urinalysis and give your pee, they would expect to see these inside, this nitrogenous waste. If they see cells in there, or platelets, or some plasma proteins, it becomes a problem. It means that your kidney is having some issues. Again, I put all of the stuff here, similar thing, talking about, again, the filtration, tubular reabsorption, and then secretion. Again, some of the content that we have, I said, put here the reabsorption of material, amount filtered per day of water, amount of sodium, glucose excreted. We don't want to secrete. If your urine tests sweet, not that you should go testing your urine, but if glucose is secreted, if they find glucose in your urine, it can be some problems. And then, but people who are diabetic too, they try to get rid of glucose, and some of it comes out in the urine. So the urea, the uric acid, most of it, all of it is secreted. So I put this other summary slide here, said secreted material into the tubule. We have drugs like penicillin, some of the ions. And if there's too much acidity, if your blood is too acidic, some of the hydrogen ions are secreted and then excreted, things that are secreted into the tubule. Now you see this is the stuff that is secreted. The things that would influence what is secreted or how well the nephron works. If you have hypertension, high blood pressure, if you have diabetes, the diet you are on, some medications would affect the way what is being filtered, because some of the medications you take, like after you are taking your own antibiotics, some of them would be secreted out through your kidneys. If you take penicillin and you go pee, your pee will smell like penicillin. If you have hypertension, means that your kidneys are holding up more sodium. When they have more sodium inside your blood, then it pulls in more water. So your kidney's job will be trying to get rid of as much sodium ions as possible. Diabetes, if your blood glucose sugar is too high, your kidneys might say, let me help out here. And instead of reabsorbing all the glucose, some of it would be released. I say healthy kidneys will filter out about 180 liters of blood per day. Now, I think let me just step back here and let's take, before we look at the other part here, let's take a quick look here about urine formation. Hopefully it works.
The renal system consists of the kidneys.
Can you hear? Can you hear? Okay, great, fantastic.
The connecting arteries and veins, and the urinary tract. The kidneys are paired, bean-shaped organs lying outside the peritoneal cavity in the back of the upper abdomen, one on each side of the vertebral column. The kidneys function as the body's main excretory organs, eliminating the body's metabolic waste products by filtering the blood. Substances that are unneeded or are present in excess are filtered out of the blood and form into urine, moving via the ureter into the bladder before being expelled through the urethra. The kidneys also selectively reabsorb those substances that are needed to maintain the normal composition of the blood. By adjusting blood composition, the kidneys are able to maintain blood volume and pressure, ensure the proper balance of sodium, chloride, potassium, calcium, hydrogen, phosphate, and pH, and eliminate products of metabolism such as urea, uric acid, and creatinine. The medial border of the kidney is indented by a deep fissure called the hilus, where the blood vessels, nerves, and ureters connect to the kidney. The kidney is composed of up to 18 lobes. Each lobe is composed of nephrons. Nephrons are the functional units of the kidney, and each kidney contains more than 1 million nephrons. Each nephron consists of a glomerulus and a system of tubules. The glomerulus is a unique, high-pressure mass of capillaries that filters the blood. The glomerulus is encased in a thin, double-walled capsule called Bowman's capsule. The space inside the capsule and surrounding the glomerulus is called Bowman's space. Plasma-like fluid is filtered from the capillary blood into Bowman's space through the glomerular filtration membrane. The glomerular filtration membrane consists of 3 layers of capillary wall: the endothelium, the basement membrane, and the epithelium. This membrane allows some particles from the blood to pass through, but not all. The fluid that is filtered from the capillary blood into the Bowman's space is called filtrate and forms the primary urine. The filtrate then diffuses across Bowman's space and into the tubule system of the nephron. In the tubules, some substances are added to the filtrate as part of the urine formation, and some substances are reabsorbed out of the filtrate and back into the blood. The nephron tubule is divided into 4 segments. The filtrate passes through each of these segments before reaching the ureter. A highly coiled segment called the proximal convoluted tubule, which drains Bowman's capsule and where almost complete absorption of nutritionally important substances takes place. A thin looped structure called the loop of Henle, which reabsorbs water and ions from the urine and plays a role in controlling the concentration of urine. a distal coiled portion called the distal convoluted tubule, which regulates potassium, sodium, and pH, and where further dilution of the urine takes place, and the collecting tubule, which joins with several tubules to collect the filtrate and where final sodium regulation takes place. Each kidney is supplied with blood by a single renal artery that arises on its respective side of the aorta, before dividing into 5 segmental arteries that enter the hilus. Within the kidney, each segmental artery branches into several lobular arteries. The lobular arteries further subdivide to form interlobular arteries, which branch off into afferent arterioles. Blood flows into the glomeruli through the afferent arterioles. Blood flows out of the glomerulus through the efferent arteriole. The afferent and efferent arterioles regulate glomerular capillary pressure by selectively dilating or constricting. The kidney's venous blood, now filtered, flows from the glomerulus via the efferent arterioles into the peritubular capillary network, a low-pressure, reabsorptive system surrounding all portions of the tubules. This arrangement permits rapid movement of solutes and water between the fluid in the tubular lumen and the blood in the capillaries. The peritubular capillaries rejoin to form the venous channels by which blood leaves the kidneys and empties into the inferior vena cava. Urine formation involves the filtration of the blood by the glomerulus to form an ultrafiltrate of urine, the tubular reabsorption of electrolytes and nutrients needed to maintain the constancy of the internal environment, and the secretion of waste materials. Filtration occurs as blood flows into the glomerulus from its afferent arteriole, and plasma moves through the glomerular capillaries into Bowman's space. From Bowman's space, the glomerular filtrate moves into the tubular segments of the nephron. Here, through tubular reabsorption, electrolytes and nutrients move from the filtrate back into the bloodstream. Here also, through tubular secretion, substances move from the peritubular capillaries into the urine filtrate. The filtrate concentrates in the collecting tubules, then finds its way to the renal pelvis, where it is directed to the ureter, the bladder, and the urethra for elimination. The kidneys perform an excretory function by filtering the blood and then selectively reabsorbing those materials that are needed to maintain a stable internal environment. The nephron is the functional unit of the kidney and is composed of a glomerulus, which filters the blood, and a tubular component, where necessary substances are reabsorbed into the bloodstream and unneeded materials are secreted into the tubular filtrate for elimination in urine.
Okay. So, does anyone have any question? Any question? Okay, let's just finish this section and then be done with this. So I said, how is blood volume and pressure maintained by the kidneys? We said reabsorption of salts increases blood volume. Reabsorption of salts increase blood volume. So we said aldosterone, a hormone, promotes the excretion of potassium and the reabsorption of sodium. When they're talking about reabsorption of salt, the higher amount of sodium you have inside your blood, the more it would do through osmosis would make water to come into the blood, into the blood to increase the blood volume. Atrial natriuretic hormone secreted by the heart when the blood volume increases and inhibits the secretion of aldosterone, which promotes the secretion of sodium. So you have a situation where you have these hormones here that would make more sodium to be secreted or more sodium to be reabsorbed. In just playing this kind of balancing, controlling the amount of sodium in the blood, it helps to maintain the blood volume. I talked about establishing a solid gradient greater than towards. So the point, if you want to understand how all of this water is moving, sodium is moving in and out, is great. But I just want you to know that in terms of the kidney, like this, within the nephron level, it controls how much amount of sodium is eliminated and how much of it is retained to influence the amount of blood volume, hence blood pressure. And we notice that this aldosterone is involved. Alcohol, we know when you drink alcohol, you pee a lot. The alcohol inhibits antidiuretic hormone. And when it inhibits this hormone that is this idea of antidiuretic, means it stops us from from peeing. When it inhibits that, then when you drink alcohol, you end up peeing a lot. If you are thirsty, the worst thing to do is to drink alcohol, because when you drink it, the alcohol part of it would inhibit this hormone that makes us not to pee, and then we end up peeing a lot and you lose water. I talked about how the kidney controls the pH. You can look at that later on. Now, when we want to pee, it's a different story. So this is our bladder. This is where the ureters are coming in, and then this is our bladder. When urine fills up here, it would send signals to our spinal region, to our spinal cord, and then also some signals will now leave from our spinal cord and say the bladder is full, go now to the sphincter. A sphincter here is some that will, when stimulated, they will contract, and then this opening here will be open through the urethra, and then we pee. For babies, they have the situation where it fills up, the signal gets here, it goes here. This one causes this to relax, and then this one, and then it contracts. This opening happens, the baby pees. So the baby pees anytime it happens without the baby really knowing. As the baby grows and gets bigger, the signal now goes right to the brain. There's a center in the brain that would receive the signal when the expansion of the walls of the bladder are sensed. The signal comes here, and then it goes to our brain. Our brain goes there and now goes and makes this signal to come here and said, listen, do not, if it is not proper, this is not a good time to pee, and then you are able to control it. So for babies, they don't have this connection to the brain to influence how we pee. So it's more like pee is full, signals come to the spinal cord, automatically they cause contraction to happen, and then they pee. But for us, when you grow to be an adult, the signal comes to your brain, you are able to send signal to this sphincters here, say, this is not a good time, don't pee in the class or something like that. And then you say, hold it, then it stays until when you are ready to go out, take a break, and then you're able to go to the bathroom to be able to, and then the signal will go, okay, now it's okay for you to allow this urination to happen. I mentioned that urinalysis as a characteristic of normal urine. Urine usually has a pale yellow color. However, may vary from colorless, dilute to dark yellow if concentrated. Abnormal findings in urine, like when you have glucose inside, when you have certain proteins in there, certain nitrates inside, and nitrites, or you see white blood cells inside. Those will be conditions that could inform that you might not be in good health. That is why they normally, when you have certain symptoms, they will ask you to give a sample of urine for them to analyze it. I said diabetes, when you have blood glucose is above normal and then it appears in your urine, that can become an issue. Sometimes when people we don't have this glucometers to see whether you are diabetic or not. When people pee, some people will pee outside, and then their pee would attract like bees there, because bees of the sweetness that is present in there And so, because normally we are not supposed to have glucose in our pee, and people who have diabetes, one of the symptoms that they have, trying to eliminate some of the glucose, they end up peeing a lot. So some disorders that are involved with the kidneys will be diabetes. We mentioned about hypertension. Diabetes, some of it can be inheritable. Hypertension is also, some of it is inherited, and they could end up having big issues for the kidneys at the level of which you have your kidneys failing totally, or sometimes you might also have infections of the urethra, which is the passage outside I mentioned earlier. And then you could have a bladder infection, when an infection of your bladder, or sometimes you could have the nephrons themselves getting infected, polynephritis, or you could have your urine, you don't have it's so concentrated with certain ions that they form kidney stones, and then when the kidney stones happen, they can form blockage and could form a whole bunch of other things. And then uremia, say high levels of urea and waste substances in the blood that, because they are not being removed from the blood properly, causes serious conditions. And water and salts are retained because the nephrons are not working properly. When your kidneys are messed up, then you could go to a dialysis center. What does a dialysis center do? Just in the same way as which your afferent arteriole is bringing blood to your kidney, and then the efferent is taking blood away from the kidney. When you go to a dialysis center, they hook up your artery to this machine. It's almost like and then it will do like similar to what the nephron is doing. But this one is not like many machines. This is one big machine. And then the dialysis is happening here. Dialysis happens when certain molecules are allowed to come out, and then others are not. And then, but there is a contraction that enables us to do filtration of the blood outside of your body, and then it brings it back to the body as well. And then sometimes you could have a situation where this, normally you would go to a dialysis center, but now sometimes they have portable systems which would allow people to carry on them and be able to go around their business without going to spend several hours at the dialysis center. And then better yet, sometimes people can have the kidneys replaced from a donor that that matches them, and then the science has been able to improve such that they can transplant kidneys easily. Now I said again, our kidneys help to eliminate waste in terms of function. They maintain water salt balance, acid balance, and then the other, because they also help, like for example, producing erythropoietin. They'll help our skeletal system and our cardiovascular system. And then because if they produce erythropoietin, they will produce helps when the production of red blood cells, right? And then we have they also help our digestive system and our nervous system. All of these things work together to maintain a good homeostatic state for us. And then I kind of put here the various interactions between the different systems. As you can see, the kidneys are receiving blood, so the cardiovascular system is involved. The digestive system is producing material that goes into the liver and is carried is absorbed in the blood, taken to the liver for breakdown, and then when it breaks some of the material in, the separation of the wastes happens in the kidneys. Our muscular systems are there to enable the movements of some of the blood through our veins. So we have our nervous system sensing things and telling our kidneys and our bladder to be able to pee. And then we have oxygen. So we have all of these systems again The point is trying to work together to make sure that as an organism made of organ systems, we are operating properly. I'm going to stop sharing here. Remember, we talked about our red bone marrow forming, giving us secret, helping to make our blood cells. So let's just go ahead and look at it. I said, so by the time that we are done, I would like you to think about the functions of the skeleton, some of which we have discussed. And then you will look at the different parts of some of the different types of bones that we have. All our bones are not the same. And then we'll look a little bit more at a bone that we refer to as a long bone reference because of its shape and how it looks like. And then we'll talk about how bones grow, what happens when we have the bones are messed or broken, like in a fracture, and they have to be repaired. And we'll talk about some of the disease conditions that happens as a result of something going on with our bones. And since bones attach to other bones, we'll look at some of the joints at which the attachment happens or where they move around, because they're not really attached per se, but they are moving around. Some of them move at the ends around certain junctions we call joints. So in general, I would say we have our bones together referred to as the human skeleton. They usually start originally in like cartilage, meaning not like strong bone. If you look on if you eat ribs, by the end of ribs, they have the whitish stuff there that is cartilage. If you eat a drumstick, when you eat a drumstick, at the ends of those bones, you see the whitish stuff there that is cartilage. I said when we start forming as a fetus, when the bone structure that is forming is usually in the form of cartilage, a particular type they call the hyaline cartilage, which is also the most important. And then when we start developing, when you are born, some of your bones have been impregnated with calcium and phosphate to form strong bones. But when you are born, some areas, like even in our skull, the bone, some of the areas are still in the cartilaginous form. But by the age of 25, everything has hardened up because of the impregnation of calcium and phosphates to make the bone strong. Our skeleton generally is made up of 206 bones, and they account for about 20% of our body mass. 80 of those bones, we call them the axial skeleton. That means if you look at like our spinal cord and our neck and our skull, all those bones and our ribs, we refer to those bones. That means our spinal cords, our ribs, our rib cage, and then our neck, and then our skull, like the axial skeleton, that is made of 80 different bones, and the rest of 126 are called appendicular skeleton. They are attached to this axial skeleton. In terms of function, we said they provide support. They provide support of the body, and then they provide protection to soft tissue. Our brain is soft, our heart is soft, our lungs are soft. They produce blood cells because at the end of the red bone marrow, we have stem cells there that would produce that. They store minerals, calcium and phosphate. That's what goes and impregnates the hyaline cartilage to make it strong in the begin with. In the long bones that have hollow material inside, especially, they have fat marrow, bone marrow in there that has mostly made of fat. And then when the bones are connected to our muscles, they are attached to our muscles, then they enable for movement because we as a support system, and being able to move around, our bones help us to accomplish that. In terms of how we classify bones, we can classify them based on the basis of shape. Remember, we're talking about long bones. So,
Professor,
yes.
Sorry, I don't mean to interrupt. I just don't know if you're supposed to be sharing, but we don't see like the first few slides.
Oh, that's fine. Thank you for saying that, because I've been advancing the slides as I went along. So sorry about that. My excuse. Okay, let's do that. Let's do this. Okay. Okay. Take 2. This is where we were. I mentioned how the bones grow, but all of this material in terms of function, this is where we were. This is where we should look at. I said all our bones, we have about 206 bones. We said you can divide them into axial and perpendicular and appendicular cartilage, skeleton. The axial ones, I said, had to do with the spinal cord, vertebral column right here. And then you have our ribs that are connected to it, then our neck bones, and then also our skull. That's referred to as the axial skeleton. And they make there are 80 bones involved in this mix. Then you have the bones that are attached to this, called the appendicular skeleton, like the ones from our that as collarbones, and are the ones that are attached to our arms and all of this, and our leg bones. I said together, all our skeleton makes up 20% of our body mass. I said at the beginning, when we are forming, all the bones we have as a fetus, they come up in the form of cartilage, which is softer than compact bone. And then when we are born, some of them have converted to compact bone, but some of them until the age of 25 are not hardened. But at the age of 25, everything that was supposed to be impregnated with calcium and phosphate to make it hard bone has been hardened up. And then the rest of the other bones that remain as cartilage, like the end of the long bones that you have cartilage there, those will remain as cartilage. Then I went on to say here, in terms of function, so most of which you guys talked about: support, protect soft tissue, produce cells, store minerals, and I meant calcium and phosphate, because that's the material that is made of the compact bone. Then it has also stores fat in the bone marrow. You hear red bone marrow found at the end of the long bones, and yellow bone marrow found in the shaft of long bones. In terms of classification of bones, but you can classify them based on shape. Like you could refer to these ones here, bones here, are long bones. And then the ones at our wrist area, they're irregular in shape. So they're kind of short, like round and stuff. We call them like short bones. And then if you look at the ones that make up our vertebrae, they're kind of irregular in shape, so they just call them irregular bones. The ones that make our skull, they look like sheets that were kind of bent to fold to form like a round shape with connecting with each other at suture lines. So they call these flat bones because of their thin nature. So if you look at the long bone, which I said we'll talk a little bit more, is that it has at the end here, it has spaces. It's not, if you compare this area here, the shaft, it is compact, it's thick. But at the end here, you have bone that forms like, it's almost like a scaffold where you have like holes inside the area. So this is the area that is referred to as spongy bone. And it is in this spongy bone area here where you have red bone marrow. And it is this red bone marrow that has stem cells that would would go and produce blood cells. So you have here, you have this shaft area here. It has yellow bone marrow, which is mostly fat. Then I mentioned the bones of the wrist that are referred to as short bones, and then the bones of the flat bones in the skull, and then the ones that are on a vertebrae irregular because of their shape makes it look like that. And then if you look more now in the long bone, this area here that has the spongy bone is referred to as the epiphysis. And then this area here, which has the compact bone, is made of the diaphysis, the shaft again containing yellow bone marrow as well as blood vessels. Then if you look at it, there is an outer covering, even though our bones look like, if you know, you have not seen anybody, I can always liken them to the bone of, I don't know, like a cow bone or something like that. There is an outer covering here on this long bone that is called a periosteum. It has fibrous connective tissue. And then I said here, fibrous connective ligaments connect bones to other bones. Blood vessels in here, yellow bone marrow here, some blood vessels here, more spacious here, spongy bone, red bone marrow with stem cells that help in making blood cells. If you take a microscopic view of the cross-section of the long bone here, it almost looks like a tree with rings, but the rings are not for the tree. The rings will be around the entire the entire stem of this tree if you're going to cut it. But here you have these smaller rings structures called the osteons. And within this ring structure that calls the osteons, in the middle of it, you have these blood vessels. And from the blood vessels, you have a concentric ring, an internal ring, an outer ring, and then the next one. But within these ring structures, you have cells called osteocytes. So osteocytes are living bone cells. And then I said this structure here is called an osteon. So you have, in a compact bone, you have many osteons in there, and each osteon containing osteocytes. As you're going to see, when these bones are forming, like out here, if this bone is forming and it was going to get bigger, now one of these bone cells, which is still developing, called an osteoblast, would start creating a ring structure around a blood vessel, and then afterwards it will lay more connective tissue and calcium and phosphate deposits there to surround the blood vessel to form an osteon. We'll look at that in a few minutes. So osteocytes are mature bone cells. Osteoblasts are cells that are growing to eventually become mature bone cells. So I put here again, what are the three types of cartilage found? I mentioned that there is, before all of us started, like cartilage, which is more flexible than compact bone. We have, depending on how flexible the cartilage is, they have different types of them. We have three different kinds and located in our body in various areas. Hyaline cartilage is the one we found at the end of long bones, in our nose, at the end of our ribs, and then in the larynx and our trachea. All that is hyaline cartilage. In between our vertebrae, like our backbones, all of those irregular bones in between them, we have fibrocartilage. We also have it in the knee. And in terms of strength, it's more stronger than the hyaline cartilage that is much more, that is not as strong. Then the elastic cartilage we find on our ears and our epiglottis, things that could wiggle more, is more flexible. If you look, you can make the pinna of your ear to wiggle. And the epiglottis we saw can go to cover our windpipe when we are swallowing food and back and forth. Those are more flexible, but all of these are cartilage. In terms of the bone cells that I was talking about, you have osteoblasts, which are the cells that will create more bone to create more osteons. Osteocytes is when those cells have matured within the osteon. And then osteoclasts, when let's say, for example, we want to, your cells, your bones want to grow, the bones are either at the edge of at the tips of for the long bones, for example, some of it will be reabsorbed for the osteoblast to go there and try to extend, grow more bones, osteons there for it to grow taller. And then chondrocytes are the bones that form cartilage. So kind of notice osteoblasts, the bones that go to initially increase, or they either involve in repair or growth of bone. Osteocytes are the mature bone cells that come from the osteoblast. Osteoclasts are the reabsorbing bone cells that would need to break down and then to allow for osteoblasts to come there and extend the bone or repair the bone. Then chondrocytes make up cartilage. So then the other thing we should also consider is how I mentioned forming bone. There are two ways in which bones form. They have fancy names for them. They call, one of the names is intramembranous ossification. Intra, in between, membranes, layering of bone. So the bone develops between sheets of fibrous connective tissue. If you can imagine that you have flat bones, the bones of our skull, so you have sheets of connective tissue, and then those bone cells would go there and they layer the bone there, and calcium and phosphates would make them stronger. That's called intramembranous ossification. Then endochondral ossification is when the cartilage that was formed in the shape of the bone is now replaced by bone cells and calcium phosphate to make them strong. So if you take each one, I think I have a picture of it somewhere. Let's see. Okay, let's first go to endochondral ossification. I said the first step is the cartilage model. The chondrocytes lay down hyaline cartilage in the shape of the future bone. I mentioned that when we are beginning at all our skeleton, our structure, the structure of our skeleton is in the form of cartilage. So if you look at endochondral ossification, the process starts by laying down of cartilage in the shape of the future bone. Then next, I said osteoblasts secrete bone matrix and results in compact bone being formed. So I said primary ossifications, osteoblasts are brought interiorly by blood vessels and lay down the spongy bone. And then secondary at birth, for example, bone centers in the epiphysis at the ends of the bones formed after birth, epiphyseal plates. We talk about the cartilage band that acts as growth plates that allows the bone to lengthen. I think I have, imagine that your long bone, well, as a fetus, this is the bone that will, when you grow up, is going to be big. When the baby is born, it's going to be this big, the long bone, but it starts in this form. We call again the ends here are epiphyses, the middle center are the diaphyses. So cartilage, chondrocytes will lay out this shape. Osteoblasts will come now, and now we start increasing in this area of the diaphysis. I said blood vessels bring osteoblast. They are the blood vessels that bring this into this area to make for them to start doing what they do. And then I said the medulla cavity, this area cavity forms, and then the edges here thicken up. Ossification happens when the calcium and the phosphate starts creating compact bone here. And then you'll see here at this end here, as the cells here mature, as osteocytes, these ones here are still the osteoblasts that will be involved in extending this bone. Sometimes at this interface area here, you have osteoclasts that will break down some of this bone that is strong here to make sure that it allows for the osteoblast to come and start extending from this end and from this end, compact bone. Then at the end of the day, you have this cartilage here. Here you have compact bone here. Inside here, you have blood vessels along with yellow bone marrow. But meanwhile, we have red bone marrow here. I think, let me look at this. Let's look at a short video here on how bone forms around a blood vessel. And then the next, we will also look at the situation, osteoporosis. We can watch that later on, but let's take a peek at this one, an animation of it.
Bones grow by forming new bone on the surface of older bone or cartilage. Osteoblasts deposit an extracellular matrix containing calcium and phosphorus salts to form ridges around a blood vessel. The groove in which the blood vessel lies is transformed into a tunnel when the growing adjacent ridges of extracellular matrix meet. Osteoblasts present at the inside of the tunnel deposit extracellular matrix, forming a new layer of bone. A new osteon is formed containing osteocytes in chambers arranged in concentric circles around the central canal.
So I mentioned the idea that you have this is a long bone here, and then you would see, they said this is the diaphysis, which is the middle section here, but you have this area here where they call it the degenerative zone. This is an area where osteoclasts will break down this area, and then the lengthening, where this proliferation of osteoblasts starts dividing, and then the lengthening occurs to be able to extend from this ossified region. When this is absorbed, more ossification now extends, and then the bone gets longer. There are some hormones. What drives our bones to grow? We have the growth hormone that stimulates, this idea of reabsorption of the bone at the ends for them to be able to lengthen. The more growth hormone you have, the more that happens. Sex hormones also affected, because at adolescence, your bones, like when they say sex hormones, what are they? We're talking about like testosterone, estrogen, and all of that. They influence how our bones grow as well. Then vitamin D is also crucial because it allows, if you don't have it, calcium that is required to make your bones stronger cannot be absorbed even if you eat it in your diet. But in the presence of vitamin D, you are able to absorb it, and that will make your bones get stronger. I said normally that we have something called bone renewal. Our bones keep doing this regeneration to make sure that they get stronger. About 18% of the bone is remodeled each time. And when for our bones to get also stronger and remodeled to get stronger, the more pressure we put on those bones, the more stronger they can be. I said it also there's also regulation of calcium in the blood through. Okay, imagine that as you're going to see in the nervous system, calcium is super important for our nervous system to work, which is our nervous system is like the AT&T or the Verizon that connects, allows communication between different parts, between different cells, sending how in the network that messages get sent through. Now, you would see that calcium is very important in that process. And the way we get calcium is that we eat it. We don't make calcium. We get it from our diet. What happens if you don't eat a diet that has enough calcium? Your body will prioritize you getting strong less than the need to communicate between activities in your nervous system to work. So for able to maintain the nervous system, to keep it online, the calcium that is required to make it work, our bones, it would leach out of the bones to supply greater availability for our nervous system. So we have hormones that sense the amount of calcium in the blood, either remove some of it and deposit it in the bones, or remove it from the bone to add it to the blood to make sure that the blood volume is fine. So we have parathyroid hormone, which increases blood calcium level by causing bone recycling, making the calcium to leach out of blood. And then calcitonin, if in our diet, we out of a sudden, we bump up, we had something that had a lot of calcium, is increasing our diet, calcitonin would remove the calcium from our blood and deposit it in our bones. When for some reason, the things that make our bones remodel and get stronger, they are not happening very well. And then we also need, our intake of calcium is also less. So you would see parathyroid hormone happening where it is removing calcium from our bones to put it in our blood. And then the result is that your bones can become weak. Sometimes they can become, have the condition that they refer to it as osteoporosis. So let's look at that condition, and that ability to lose bone mass because of leaching of calcium. Sometimes we're seeing that if you're sex, if you're a woman, you are more likely to have that. If you are if you are Caucasian, if you are Asian, if you are thin, if you have a family history, if you have early menopause, there are many factors that here could cause somebody to have their bones leaching out calcium, making their bones to be weak. And when they are weak, guess what? They can break easily. Somebody pushes you, you fall, you have a fracture. Fracture is when the bone gets broken. And then I said it can be treated with some drugs, hormones, and lifestyle. But let's take a look at what osteoporosis looks like.
At around age 30 in humans, bone loss becomes an issue. The process of remodeling bone slows, and the loss of bone calcium deposits exceeds its replacement. Beyond this point, it is currently no longer possible to substantially increase bone density. Over time, the density of the structures of the bone diminish, which leads to general weakening of the bone structure. It is a very gradual process and requires decades before any noticeable bone weakness appears. Osteoporosis is the end result of the erosion of the structures of the bone. Osteoporotic bone has much larger spaces in the bone structure and is significantly weaker than normal bone. Therefore, the bone is more likely to fracture. This leads to many serious injuries in the elderly due to falling that severely impact their mobility and health status. Women are more susceptible to osteoporosis than men. This is probably related to hormonal influence on bone growth and calcification. Also, deficiencies in dietary intake of calcium and other nutrients, including protein, phosphorus, vitamin A, vitamin D, vitamin Km, magnesium, zinc, and copper, will negatively influence bone density. In addition, genetics have an influence on bone density. Strategies for preventing osteoporosis in women include meeting their dietary needs for calcium, vitamin D, and other nutrients. Physical activity that includes weight-bearing exercises is important to maintain bone mass. Postmenopausal women have in the past been prescribed estrogens, estrogen receptor modulators, and calcitonin as medical treatments for osteoporosis. These treatments are under review now. Calcium supplements are still recommended, but regular exercise, including weight-bearing and resistance activities, are proving more effective. Finally, since both tobacco smoking and alcohol consumption have negative effects on bone growth, these behaviors should be avoided for healthy bones. In summary, osteoporosis, the weakening of bones due to loss of calcium and phosphorus from the interior of the bone architecture, is a degenerative disease that is influenced by many contributing factors. Health-promoting interventions, such as adequate nutrition and physical activity, coupled with positive behaviors such as not smoking or using alcohol excessively, can forestall the onset and severity of osteoporosis. Men are also at risk for developing osteoporosis. For men, similar prevention strategies apply.
So when we talk about osteoporosis, if you think about it, that the way to treat it, sometimes they will give you calcium supplements, sometimes they will give you hormones for women. One of the things that you sometimes when you look at conditions like this, you can also imagine that before they came up with Western medicine, people did not have access to hormones and calcium being given to you. How did the people before this time, how were they able to maintain strong bones? One of the things that we share with the people of those days was that we have a choice. We can exercise. That means we can move around and carry things or do things that are weight bearing. And of all of these things, when you take supplements, whether they are hormonal supplements or calcium supplements, some of these things have, especially hormonal supplements, some of them might have some side effects. Among all of the things that will not have any side effects at all would be the physical activities you do or the disengagement of some of the adverse behavioral activities like smoking and alcohol. So before you start looking at, okay, maybe I'll just take hormones to be able to resolve this, I think my greater takeaway from this is exercise more. You have control over that. Avoid smoking and alcohol consumption, especially sometimes smoking has zero benefit. We used to even think that alcohol has some benefits, but recent studies are almost making the benefits of any amount of alcohol to be negligible. But all of these behavioral activities, we have control over. But the other thing too, again, is with the information ability to genetically test, because we say that your gender and your genetics can inform whether you are susceptible to have osteoporosis. The good thing about it is that today you can actually test to find out whether you have a potential or at risk of having osteoporosis. So we've talked about this, but let's look at another situation here. This one was going to talk to you about what the osteoporosis, which we just saw. And then here, what happens if you play soccer or you play some sports, sometimes you could have a fracture. So when you have a fracture, when your bone like breaks into two, let's look at what happens in the healing process of that bone, because they do heal. When you break your bone, I would just go to this picture first. This guy probably was playing soccer, then out of a sudden, we know he broke his bone. We know the bone has some blood vessels, but not as many as you'll see like on the skin, but it has blood vessels. So in that breaked area where there's a breakage, some of the blood will be leaking out in those areas where the blood vessels were messed up. But as they are being broke, as they are coming out, the healing process too would start coming into gear. Some things the clotting process that we talked about would be involved in having to stop to stop the blood from oozing out. And then these blood vessels would be carrying something called angiogenic factors that will make them to grow towards each other and connect. But relevant to what we are talking about is that they will also bring about chondrocytes to come and lay down cartilage in this area. So if you imagine, when you have a fracture, when you go to the hospital, they don't really do too much. They just kind of look at is it a crack that is a clean crack, or does it have some chips? Is it chipped into little pieces? If it is a clean crack, a cut, basically what they do is they take this, they call it plaster of Paris. Basically, they take it's like a bandage that when they soak it in water, they wrap it around the area. When it dries, it hardens up. So in this case, like this guy is wearing it right now, it hardens it, maintains hold the bones together so that even though he is walking, it will not change any pressure on the fractured portion, whether the top or the lower, it just kind of hold everything in place. During this period, the cartilaginous callus, they call it, laid down by the chondrocytes would form. And after they have been formed, then now the bone forming cells the osteocytes would now come in and the osteoblasts will now come in and start forming osteons around the blood vessels. And then afterwards, the osteocytes, the mature blood cells would form, and then the healing in a several months, a few months, everything will be restored. So hematoma, 6 to 8 hours, happens in the area where there's blood forming, and then you want clotting to happen. Fibrocartilage callus, lay down. Bony callus with the osteocytes, or with the osteoblasts coming there, adding them, and then the remodeling happening when the osteoblasts start forming around the blood vessels to create new osteons to connect the broken parts. as a general speak now, we talked about the number of bones. We have 206 appendicular, axial skeleton, and then the appendicular skeleton. What do I expect you to know from this? Do I expect you to know all these bones? No, I will expect you to know for our class just the number of bones. And then, but if you're going to take an anatomy and physiology class, you will be expected to know all of these bones. So the science focus, I said, okay, what can we know from our skeleton? From the skeleton, you could determine and have a good idea of what the age of that individual is. You can also, from the pelvic bone structure, you could determine the gender. And sometimes you can determine the ethnicity, but it's a little bit much more difficult. If you look at all of these structures here, this is what people used to use early on before the molecular detection or determination of individuals like DNA they were using like bone structure and all of that. But now, if they look at somebody's remains or something like that, they could determine who you are or by your age and based on your DNA. So we said the axial skeleton, which is that middle portion, middle of your skull. There's one bone called here, this hyoid bone. It's the only one. Most of our bones are connected to other bones. The hyoid bone is the only bone that we have that is not connected to any other bone. Well, I'll show you that in a bit. And then part of the axial skeleton, the vertebrae we talked about, and the rib cage. The skull. Do I expect you to know all of these parts? No. Usually referred to as a cranium. It does what? It protects the brain. We're talking about the function of it, of the skeleton as protecting soft tissue. I think the brain is a critical soft tissue that needs to be protected. Then part of it, you have those ones that cover our face, the back. But I said this hyoid bone, the one on our neck, if you go to the doctor's office, they will touch it and say, swallow. When you swallow, it would move. Then they can feel it. That's the only bone that does not articulate or that does not connect with another bone. The vertebrae, we have 33 vertebrae, all of these bones. And one of the things that you will see is okay, let me ask you differently. What do you think is a benefit of us having the cartilage between the bones of a vertebrae? What do you think might be the benefit? For cushion, exactly. And because when this cartilage too is not there, there are nerves that the bones start rubbing against each other. That can be a big time issue. Any other benefit? If you notice, it allows us to be able to also bend. I can sit here and go this way, lean forward, go backward. All this flexibility, the ability to do all of those moves is, if all of this was just, there was no cartilage, it was just a rigid connection of hard bone with a spinal cord inside, it will be rigid in this shape. But the fact that you have this cartilage in between the vertebral disc, it allows us to be able to move in different directions, and I think it's a benefit. And as she said, it provides us some cushion, and also then protects the nerves that are inside. Then, as part of that axial skeleton, we've talked about the cartilage. We know the part it plays. We've talked about the rib cage, the ribs. We talked about the part that it plays to protecting our heart, our lungs, And then the fact that you have cartilage in connecting these stronger bones here, cartilage, again, we know it allows for movement. When you inhale, your chest rises, the rib, the muscles in between the ribs, the intercostal muscles would contract. When they contract, they pull your rib cage up and out. And if everything was rigid, this idea of moving up and outwards might be a little bit challenging, but because of the flexibility of the cartilage connecting to these stronger bones, to the sternum, which is the mid portion here, it allows us to be able to breathe and then our chest to rise and fall. And then we said from the axial skeleton, you have the appendicular skeleton, you have the pectoral girdle and the upper limbs and the pelvic girdle and the lower limbs. So you have all of this stuff. So these are the bones that would articulate, connect with the axial skeleton. These ones of our arms and the ones from our legs. Do I expect you to get to know all of these ones? No, but I would like you to know that these ones, the upper, the ones from our arms and the ones from our legs do connect. And I put these ones here again. If you are taking this class to eventually take anatomy and physiology, you might start even just trying to memorize this. So we said, for example, this bone here, the pelvis connects to this one here through some kind of a joint here. And we have different kinds of joints. This joint here that is connecting this one to this one is different from the one that connects the plates on our cranial bones, like the bones that are in our skull. So I will talk about different kinds of joints. So we have fibrous joints that are usually immovable. So the plates, the flat bones that make up the skull, they are connected almost like interlocked with each other. And when they're interlocked in that fashion, those plates cannot move, they stay in place, they are immovable. I said cartilaginous, they tend to be more slightly, joints that are cartilaginous tend to be much more slightly movable, such as the vertebral column as we're talking about. In between the irregular bones, they allow for movement for us to be able to bend in different directions. The ones that move a lot more are the synovial joints, where they say like a ball and socket bone. So the synovial joints allow for more flexible movements. So if you look here, I know everybody in the class can do this. It allows this one here to move around, to be able to move around here. Gymnasts, this is where they differentiate. They differ from most of us because their movement, their range of movement around of their synovial joints is much better than the average person. And so when you would hear some of the joints, if you are a gymnast or you go for physical therapy, they're trying to see whether to stretch you, they will hear words like flexing or flexion, where you move your joint to reduce the angle, extension to increase the angle, adduction, bringing the body towards the midline, abduction, moving it away, inversion, moving the sole of your foot to turn it inwards, and inversion, the reverse. So if you are doing dancing or gymnastics or physical therapy, you will hear some of this kind of stuff. So all of this referring to those words those things I just mentioned to you. So this is all what I have for us talking about our bones. Does anyone have any question?
Yes.
Okay. So the nervous system. I said, by the time that we are done with this, some of the things that we should know is to look at what is the function of our nervous system. What are the kind of cells that are involved? Our nerve cells. What kind of nerve cells do we have? What is the structure of them? We have neurons. They're actually two sets. We have neurons and supporting cells called neuroglia cells. What is the difference between them? What is their structure? How is our nerve cells? I was mentioning that they connect, they carry information. How long are they in terms of shape? Are they similar to our skin cells, for example? How do they actually carry this information? And then we will talk about how the nerve cells are connected all over our body and how they connect to our brains and things like that. And then we'll probably definitely talk about how drugs, medications or drugs in the whole might play a role in interfacing with our nervous system. Now, I said we have, there are two divisions of our nervous system. But I say in general, the nervous system's job is to receive and process sensory information which we capture, information which we sense from the outside. When I see stuff, when I see you, I am seeing stuff. That means I am sensing something through my sensory organs, my eyes, my ears, or something touches me. I'm sensing from my external environment. Or if I am thirsty or I am hungry, you cannot tell from my face, I might be a little grumpy, but from my feeling of thirst and hunger is from inside. I can sense that. So is sensing from the inside and from the outside. I said there are two major divisions. The central nervous system, as we're talking about the axial skeleton, there is a central nervous system that is made up of the brain and our spinal cord. And similar to the appendicular skeleton, we have peripheral nervous system that goes through our arms, our legs, and then our sides from that are all connecting to the all of them are connecting to our spinal cord, which in turn connects to the brain. So I said in terms of function, one, it senses information, it senses our environment. A stimuli something would stimulate. And then, so when we see something, when something jumps out, it stimulates us to be able to see something. Then our eyes would capture that information. And then it will go to our brain. Our eyes capture it. It connects to nerve cells behind, optic nerves behind our eyes. Then they will take it right to our central nervous system, to the brain, where the brain does what? It makes sense. Instead of interpret, they use the word integration. It makes sense of what that is, and then makes a decision what to do about it. So you have nerve cells that are sensory, sensing, connecting to the ones in our brain that receive it and make sense out of it. When they make sense out of it, they send instructions to certain parts of our body to do something. If you see something flying towards your eye, you are seeing it, your brain, scrambles and says, close your eyelids. Automatically, the muscles of your eyelids would close and the thing doesn't get into your eye. That one is even eventually something that is so fast that you sometimes are not even aware of it. a fly or some piece of something wants to jump in your eye is already shut before it even gets there. This idea of sensory, so you have sensory input, integration, sensory output, also called the motor response. So in terms of cell types that are involved, I said we could classify those into basically two cell types. The neurons are the cells that carry the information, the sensory that sends the information, that also act like integration, and then they're also the motor ones that carry the information from our brain to our muscles or to our organs, to various organs or glands to do something. So those are referred to as neurons. They transmit nerve impulses. They call them impulses. You might say like messages, but they're impulses for a reason why you will see why. And then they are supporting cells that actually help to work to make sure that these neurons are doing their job properly. They usually supporting cells referred to as neuroglia cells. They support and they help to nourish these neurons. They usually outnumber the number of neurons. It's almost like the king. You cannot have many kings. You have many supporters supporting the king, but the whole point here is the neuroglia cells. Their job is to support and nourish the neurons, which are fewer in number. Some of the ones that locate in the central nervous system are called microglia cells, astrocytes, oligodendrocytes. What do the microglia cells, neuroglia cells, some of them are Schwann cells, and then microglia cells, some of them have phagocytic behavior. You remember how we're talking about cells that go and gobble up bacteria? Microglia cells would have some of this capability. And then astrocytes provide metabolic and structural support directly to neurons to make sure that they maintain their shape. Oligodendrocytes, they form this sheet. So it's almost like, imagine that you have the head of a water hose, but the head of the water hose now has, instead of having one outlet, it has many branches on it. And then the hose itself, the long tube is there, but instead of being smooth, it has like insulation wrapped around it that bundles around it. But if you are thinking about it as insulation, it would have just been covered with the same material without any gaps in between. But nerve cells, our neurons have this head region with branches called dendrites, which will make it in such a way that they will spread out to be able to pick up signals from different areas. And then the long strand here is called the axon. And then this axon has these things wrapped around them. They call it the myelin sheath. And we said these oligodendrocytes are the ones that secrete this material called the myelin sheath. And the myelin sheath is not continuous. It has gaps along them. They call them nodes of Ranvier. And in this structure, it makes the transmission of impulses along the axon, this long chain, they make the impulses move faster and more efficiently. So the structure of the neuron, it has a cell body, which has the nucleus, just like a regular cell. But in addition, it has this, in the cell, the cell membrane has these extensions, almost like branches of trees, or they call them dendrites. Again, their idea is to extend the surface area that they could cover to be able to pick up stimulation, like an antenna that spreads out. And then we have these myelin sheets with the gaps in between called nodes of Ranvier. And then the other end of the neuron it ends up in these branches here, but the branches have these round structures here called the synapse. So impulses would move from here and pass through here and move through this axon here, and then they end here. From this point on, they will be picked up by the next nerve cell that is here. So the question that you should start having is, how does this connection happen? How does the transmission of the impulse happen? So you have a sensory neuron receptor here. All those dendrites are there. They call it the receptor sites. They pick up the signal. It moves here. It moves here. And then it ends here again. Now, this other interneuron, which is in the brain, remember I was telling you that the brain too, that's where integration happens. It has its own neuron, is called an interneuron. If you will see that different from the sensory neuron and the motor neuron that takes information away from the interneuron itself has an axon, but absent from that axon is the myelin sheath. So all these bundles that I was telling you here of myelin, they don't have it in the brain, which is where the brain and the central and the spinal cord, they don't have this part. So impulses move from here and then are captured here by this interneuron. Interpretation happens. Information leaves from the interneuron, goes to the motor neuron, and then it takes this either to a muscle. I was talking about stuff flying into our eyes. This ends here will end at the muscle of our eye sheet, of our eyelids, for example, if we're using that example of that stimulation. And the next thing is this information jumps to that muscle and causes it to contract. So how does that information travel is what we're going to look at. Again, sensory input, integration, sensory output, the effector in this case, the muscle, but I was referring, if we're using something flying into your eye, it will not be the leg muscle, but it will be the muscle of your eye. Now, if you look on the myelin sheath, I said the myelin sheath is a lipid covering of long axons that act to increase the speed at which impulses move on the nerve, create insulation and regeneration of action potential. I say Schwann cells, which are neuroglia cells, make up the myelin sheath. Note of Ranvier are these gaps here. The mechanism by which the impulses move is not flowing through them like water will flow through a hose. They instead jump from one gap to the next, to the next, to the next. And this way in which the information moves is called saltatory conduction. The impulses, instead of flowing through, they jump. And in this ability to jump, it speeds up the rate and the efficiency in which this is done. So as you're going to see here, there are negative charges here, there are positive charges here, and then sometimes the movement, when the transmission will be involve the movement of these positive and negative charges from one side of the axon. So let's look at, and this movement is, you will look at what happens when there is not, what is the nature of the distribution of these positive and negative charges on the outside of the axon and on the inside of the axon. And what happens when a stimulus comes there and triggers the impulse to start propagating, to carry out that message that there's something has been sensed, to take it to the brain and all of that. I think this one should, let me see here. I think, let me do something here. Let me see if this works here. No, I think I have a connection that would show here. So I think, let's see. Okay, I'll go here. Watch this, how the movement happens.
Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell.
So the resting potential, when the nerve is not carrying any impulse, is the relative distribution of sodium and potassium ions. You would ask the question, on what part of the cell do we have more potassium ions, and on what side of the cell do we have sodium ions?
Is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization.
So give me 1 second, enough impulse. I don't know why those links were not active when let's copy and paste it here. It should have worked here, but I don't know why it's not working. Let me give it a take 2. Okay, I'll play it from here. Okay, let's look at this.
A nerve impulse or action potential is a change in the charge difference across a neuron's plasma membrane that travels along dendrites or axons due to ions moving through gated channels. Gated channels open and close in response to a stimulus, such as a change in the electrical charges around them. A resting neuron maintains a charge difference between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. This results in the exterior of the cell having a net positive charge, and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, and the charge difference at that location is reduced. If the change is enough, it will cause nearby gated sodium channels to open. If enough sodium ions flow into the cell, the membrane becomes depolarized. The local region becomes positively charged in the inside of the cell and negatively charged on the outside. This affects neighboring sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The sodium channels close, and the potassium channels open. This allows potassium ions to flow out of the cell, repolarizing the membrane so that the inside is again negative and the outside is positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
I have a question for you. This is a chemistry question. Both sodium ion and potassium ions are written as are positive ions. But from what we hear, we say that during the resting potential, when the nerve impulse is at when the nerve impulses when the neuron is at rest, no impulses are being transmitted, the charge distribution is such that the positives is net positive outside the neuron and net negative inside the neuron. Meanwhile, sodium ions are written as N. Let me see how. Sodium is K plus, sodium is Na plus, and potassium is K plus. How is it then that we say that at rest, the sodium that is inside is instead minus charge here, a less charge, negative charge here. Meanwhile, the potassium that is outside has a more positive charge. Who could tell me why that is so? Do you understand the question? Both sodium ions and potassium ions are indicated here as positive. But they say when the nerve is not transmitting anything here, there are more potassiums outside here and more sodiums inside here. If both are, sodium is Na plus and potassium is Na plus, is K plus, why is the charge distribution like negative here and more positive here? Who could tell me why? It has 11 electrons. So if you come here, for example, this is an atom of sodium. The blue rings here are the sodium. So you have 2 of the electrons would occupy the internal structure, the inner ring. Then the next one will occupy, there will be a total of 8, 2, 4, 6, 8. Then the last one, because there were 11, the last one would go on the outer ring here too. So now, these electrons are negative, but also sodium having 11 electrons, it also has 11 protons. So an ion of sodium happens to be written as Na plus, because for sodium to become an ion, it would give this 1 electron to somebody else. When it gives 1 electron to somebody else and you count the negative charges, there will be only 10. And then the positive charges would be now 11, meaning one more to make it be given, so be written as Na plus. Meanwhile, potassium, this is potassium. Potassium has 19 electrons and 19 protons. So but one of the things that it has in common with sodium is that they have 1 electron in the outer shell. So when potassium loses its electron to become a potassium ion, there would be 19 protons still, but there will be only 18 electrons left. And then you write potassium as K plus. But remember here that potassium has 19 protons, meanwhile sodium has only 11. So when we go back and say here that at resting potential, you have more potassiums outside here. Imagine that each potassium has 19 protons. And then the sodiums that are here, they have fewer than that, 11. So you see that if you compare the relative number of positive charges on both sides, at rest, more potassiums are outside more, and then sodiums are inside. So there will be more positive charges here than inside. That is why they say that at rest, even though you have potassium out here written as Na plus, even though you have potassium here written as K plus and sodium inside here as Na plus, there is a net more positive charges outside than they are inside. Now, when stimulation happens, then you have this situation where the potassium starts moving inside. That is the propagation of the action potential. Any question? So say the transmission of impulses along the nerve involves movement of sodium ions and potassium ions across the membrane. The sodium potassium ion, the gates open, and then you have that propagation happening. So I said resting potential is involved restoring by moving potassium ions inside and sodium outside, and then we have all of that stuff going. But my question now is, if things propagated that way, what happens when they come to the end here to connect to the next nerve cell? So, which is at this point here, what happens when they come here and they get to hand? So we've seen this action potential move right here. What happens for it to connect to go to this hand up to stimulate this other nerve cell? So what happens there is this end here, which is the synapse, there is a receptor on this side here, which is on the dendrite. You will see that has to jump. Let's see what happens. How? So at the end here, which is magnified, and then this other end here, which is the axon that is bringing it, when that action potential reaches here, you have these vesicles that contain neurotransmitters. When I said a small gap between the sending neuron and the receiving neuron, it's called the presynaptic cleft. Transmission is accomplished across this gap by a neurotransmitter. A neurotransmitter are these vesicles that are in here that contain some kind of chemical. And then when that action potential reaches here, it causes these vesicles to come and fuse at the end of this neuron and release those chemicals into this gap. The receiving neuron would have receptors on it that would allow these chemicals to come in or bind on it, and then their binding or their moving in generates another action potential to go on the receiving the receiving neuron. Calcium ions are integral in having this transmission happen. It's 1 o'clock. I'm going to stop here. And then when let me see let's watch this and then take off for now. It's not playing for some reason. Let me stop here. How about I put this like this and ask it to play? Doesn't work either, but let's watch this before we take off, please. Give me 1 second. Synapse transmission.
Synaptic cleft. The basic unit of the nervous system is the neuron, a complex cell that consists of extensions called dendrites and axons. Every axon branches into many fine endings, each tipped by a small swelling called an axon terminal. Each terminal lies very close to either the dendrite or cell body of another neuron, or in some cases, a muscle cell. The region of proximity between two neurons is called a synapse, and the neurons are called presynaptic and postsynaptic based on the direction of the nerve impulse. Though they are close together, two neurons do not physically touch each other at the synapse. Instead, they are separated by a small gap called a synaptic cleft. But if they don't touch, how is information relayed between the neurons? To better understand this, let's examine a synaptic cleft. Within a neuron, a nerve impulse transmits information using an electrical signal called an action potential. Where two neurons meet, the information must be converted from an electrical signal to a chemical signal. When action potentials travel along the axon of a presynaptic neuron to reach an axon terminal, gated channels for calcium ions, which are chemical signals, open, and calcium enters the terminal. This sudden rise in calcium ions stimulates synaptic vesicles to merge with the membrane. These vesicles contain chemicals called neurotransmitters. Neurotransmitters act as the chemical signals that relay information between neurons. The neuron in which the action potential originates is called the presynaptic neuron. The neuron that will be receiving the information is called the postsynaptic neuron. Between them is the synaptic cleft. This is the narrow space between the two cells into which the neurotransmitters will be released. Once released, the neurotransmitters diffuse across the synapse where they bind with specific receptor proteins on the surface of the postsynaptic membrane. An excitatory signal causes the receptors to admit sodium ions into the postsynaptic neuron, which results in a new action potential being generated. Overall, this process converts the electrical signal in one neuron into a chemical signal relayed by a neurotransmitter. The neurotransmitter communicates the information across the synapse to the next neuron. There are a few benefits of using a chemical signal instead of an electrical signal at the synapse. First, the electrical signals are too weak to cross the synapse, and although small from our perspective, the cellular distance between the neurons is too great. Yet another benefit is that it is possible to release multiple neurotransmitters in the same synapse. The combination of multiple neurotransmitters in the same space has a variety of functions. In some cases, the combinations inhibit the action of the neurons, while other combinations excite them. Because of this, the synapses represent the area of the nervous system where the work of processing or integrating complex signals occurs.
Cool. So, yeah, so we will stop here. I'm going to stop sharing. We will stop